Okay, so DNA replication. So this is a molecule of DNA, and DNA is unique in itself because it can create its own copy. So DNA can make a copy of itself, it can replicate. And this specifically occurs in the cell cycle during S phase of interphase. So this is where the DNA replicates before the cell cycle begins. So, to start this off, the original DNA molecule is unzipped. So, we unzip the DNA molecule, and then both of these original strands, this blue here and this blue on this side, serve as the template to create the new strand of DNA. So, the original DNA is unzipped, and then the new strand is created using base pairing rules for each side of the original strand. So this is what we say for DNA replication. It's semi-conservative because of the fact that the original DNA is unzipped, and then each of the two original strands of DNA are used as a template to create the new strands. All right, so to start off with DNA replication, we have the first step, which is an enzyme called helicase that unzips the DNA molecule. So when we say unzipping the DNA molecule, this means that the two strands are separated. So currently, what holds the two strands together are hydrogen bonds that are formed between the individual base pairs. So, for example, there are hydrogen bonds between the A and the T, between the G and the C. So the hydrogen bonds are what holds the DNA bases together. So the base pairing rules review show that A will always base pair with T, thymine, and then cytosine will always base pair with G, guanine. So this never changes even in DNA replication. So step one, DNA helicase. This is an enzyme that is used to unzip the DNA molecule. So at the location where the DNA is unzipped is called the replication fork. So this area here is a replication fork. Think of it kind of like a fork in the road. There's two paths. So this is the replication fork here. So the hydrogen bonds are broken in the original DNA molecule. Next, we have DNA polymerase. This is a second enzyme. This is going to attach to a primer. So, this primer is made up of RNA bases, and it's going to attach to the primer to start creating the new DNA strand. So, keep in mind, base pairing rules apply. So, if this were a T here, your new DNA strand would be an A. So, DNA polymerase is in charge of creating the new DNA strand. And we say that DNA polymerase only moves from the 5' to 3' direction. So, at the top of this picture, DNA polymerase is moving from right to left. And that's because the original DNA molecule here is 5' on this side, 3' on this side. So, because DNA is anti-parallel for the DNA polymerase, this is going to be 5' and this is going to be 3'. So DNA polymerase is going to move from right to left on the top because this is the 3 here. On the bottom, you can see that the original DNA is labeled with a 5' end, which means that our newly created DNA is going to be 5' on this side and 3' on this side. So because DNA helicase only moves in the 5' to 3' direction, it's going to start building randomly here, and then it's going to build out, and then it's going to jump backwards and build here. So because it moves only in the 5' to 3' direction, this is why it jumps. So the leading strand, which was the one we talked about on the top, is continuous, meaning it never jumps, it never has little fragments. So the leading strand goes in the direction towards our replication fork. So as DNA helicase continues to unzip the DNA molecule, our leading strand is going to continuously build new nucleotides. Our lagging strand, which is the strand that's going to be discontinuous, is constructed in fragments. And that's because it moves from the 5' to 3' direction. So we have one fragment here and another fragment here. So these fragments are called Okazaki fragments, and they're pieced together by ligase, which we'll talk about in a second. So just to review the leading and the lagging strand. So this is our original DNA molecule here. So if you look, there's a 5 prime on this end, 3 prime on this end, which means when we create the new strand, it's going to be 5 prime on this end and 3 prime on this end. So our top strand is going to go from 5 to 3. And then our bottom strand, since this is already 5 and this is already 3, this means that the bottom strand is going to move in the opposite direction since it's anti-parallel, and it's going to construct it in fragments because it can't move continuously, um, just like randomly in this direction. So here we have 5 to 3. And as the replication fork opens up, as more DNA molecules are unzipped, we have another 5 to 3. So these are little fragments. So our leading strand is going to be constructed continuously towards the replication fork. It never stops because as the replication fork expands, as more DNA molecules are unzipped, this will continue moving in that same direction. Our lagging strand, however, because it moves 5 to 3, it needs to jump backwards in order to replicate the DNA that continues to be unzipped. So you can pause this for a second, try drawing this yourself, and see if you can explain it yourself. So this bottom strand is our lagging strand because it is constructed discontinuously. All right. So once again, to review, your leading strand is going to be continuous, moving in the direction of our replication fork. Our lagging strand is going to move away from the replication fork, but it's going to jump backwards in order to follow through with all of the DNA replication as more and more DNA molecules are unzipped. All right, and then last step for the lagging strand only, DNA ligase is an enzyme used to attach the little Okazaki fragments together. So this only occurs in our lagging strand.